Hello, students. Let's start. Today we have two topics about pathogens of respiratory tract infections. First topic about diphtheria and whooping cough. Second topic about tuberculosis and leprosy. Now I want to demonstrate video lecture about Corynebacterium diphtheria. After then, we will discuss this topic by case study. Oh my God. Yeah, please, students, during the lecture, switch off your microphones.
अरे चाय वाला कौन है? माइक ऑफ कर लो अपना।
Thank you. Let's start. Corynebacterium diphtheria. Laboratory characteristics and laboratory diagnosis. Did you see? Right in the chat. Did you see this PPT presentation or not? Morphology of Corynebacteria diphtheria. This is club-shaped rods that form metachromatic granules, which cause Babesch-Ernst granules. Position in the smear looks like Chinese letter or Roman numbers. On the right side of the screen, you can see the microscopic pictures of the Corynebacterium diphtheria by Albert Neisser staining and Loeffler staining. You can see this is club-shaped form, which dependent is happened because this bacteria during the binary diffusion, this uncompleted binary diffusion. Next, Loeffler staining, you can see V form of this bacteria in the smear. Next picture, cultivation. For cultivation of Corynebacterium diphtheria, we should use selective media because this bacteria cannot grow on the ordinary media, and we add telluride in this agar for inhibit growth of commensal flora. And after them, we detect black colonies on this media. We can use Klauberg medium, consists the blood, and Tinsdal media, consists the telluride 2 also. Corynebacterium diphtheria has 3 biovars, gravis, intermedius, and metis, which differentiation of these biovars by morphology, cultivation and description of colonies, epidemiology activity, and different complications. Gravis morphology has short rods, uniform staining, few or no granules in the end of the rods, present polymorphism with irregular barred snowshoe or teardrops. Intermedius characterized present long barred forms, clubbed ends, for granulation, and this bacteria is very pleomorphic. Metis are long curved rods, prominent granules, and pleomorphic. Colonies on the telluride blood agar after 18 hours of incubation. Gravis forms are very small, 1 or 2 millimeter in size, grayish-black center. After 2 or 3 days, the colonies growth in size, and we detect and we can describe it like a daisy head colonies. Intermediate colonies after 18 hours of incubation, small, 1 millimeter inside and misty. After 2 days of incubation, we can describe it like a frog eggs colony. Meaties has a variable size, shiny black, and after 2 or 3 days, we can colonies became flat, center elevation, and looks like poached eggs colonies. Under the table, you can see the colonies. This is gravis, like a daisy head. You can see it. Yes, frog eggs colonies, very small, and poached eggs. You can see and shiny surface of colonies. Consistency of colonies of gravis, which looks like cold margarine. Intermedius consistency between gravis and meaties. And meaties consistency colony are soft and buttery. The toxigenicity activity of gravis is very high. Almost 100% of colonies has toxigenicity. Intermedius, a few less, 95, 99, and meaties, most kind colonies, 80, 85. Gravis have severe virulence, intermedius between moderate virulence, and meaties low virulence. Predominant strains, we detect gravis and intermedius in epidemic area. It means that not only 1 or 2 persons can be infected by these strains, and we detect it in some people. No, in a large group of people, we can detect gravis and intermedius. Meaties, we can detect in sporadic cases. And complications of this disease, if the diphtheria is caused by gravis, we can detect high fatality cases, paralytic, and hemorrhages. If we detect intermedius from the sick person, we wait high fatality cases and hemorrhages. Meaties are most characterized by obstructive lesions. Next, what the biochemical differences between gravis and meaties? What we can see, hemolysis is more characterized for meaties. Farina is present in gravis. Next, biochemical properties by different reactions. We can differentiate pathogenic diphtheria bacilli from non-pathogenic by fermentation glucose, saccharose, dextrine, presence of urease, and production of cystinase. Urease test is called Zach's test. Cystinase is called Pizu test. And what we can see on this table, diphtheria can fermentate glucose, dextrine, no produce urease, and produce cystinase. And non-pathogenic bacilli fermentate glucose and saccharose for Corynebacterium cirrhosis, doesn't produce urease, and a variable cystinase test. Pizu test. Corynebacterium diphtheriticum has positive urease and positive Zach's test, and negative cystinase test, Pizu test. This is a large table of biochemical identification of pathogenic Corynebacteria. And what we can see, cystinase is most characterized for 3 biovars of Corynebacterium diphtheria, ulcerans, and tuberculosis. Pyramidase are more characterized for non-pathogenic Corynebacteria. Farina is present in gravis, pathogenic Corynebacterium diphtheria, and cirrhosis fermentate saccharose. Urease positive test for is most characterized for ulcerans, pseudotuberculosis, and pseudodiphtheriticum. Next, what the differences between pathogenic and non-pathogenic bacilli? By morphology, we can see weakly gram-positive strains, thin bacilli, and present metachromatic granules, bulbous ends, position in the smear like Chinese letter or Roman numbers, and present of polymorphism. Diphtheroids strongly gram-positive, short and thick bacilli, few or absent of Babest-Ernst granules, positioned in the smear-like palisade, and has very little pleomorphism present. Culture, Corynebacterium diphtheria grows only on enrichment media, present telluride. Non-pathogenic diphtheroid, non-pathogenic Corynebacteria grows on the ordinary media such as nutrient agar. Biochemical test, Corynebacterium diphtheria ferments glucose, doesn't ferment sucrose, and diphtheroids ferment both glucose and sucrose. Diphtheria produce exotoxin with poisonful. Diphtheroids don't produce any toxins. And virulent tests are positive for diphtheria, negative for diphtheroids. This is testinase detection PISU test. How we prepared this test? First, we use semi-solid thin-dal media, consistent nutrient media, cysteine, telluride. We inoculate pure culture, like inject, in the bottom of the tube. Incubate in thermostat during 24 hours in 37 degrees and check results. Diphtheria ferments cysteine by cystinase enzyme, restores the metallic tellurium, and we can detect blackening appear along the injection on the media. It's a positive result. If you don't detect any changes of color inside of the media, you will check like negative result. Next, Zach's test, urease test. You should prepare the medium consisting urease and phenol red. Phenol red, if you remember, is indicator which shows us the changes of pH. If pH will change in acid condition, phenol red became yellow. If bacteria produce urease, we can detect the changes of acid condition, or we changes the pH in acid condition, and color will be yellow. Diphtheria doesn't have urease and doesn't change the pH, and the media became pink or red. Pseudodiphtheriticum produce urease and change the pH of media in acid condition, and we detect yellow color of media because in this media pH was changed in acid condition. Next test. This is about virulence, exotoxin. Corynebacterium diphtheria produce exotoxin, very poisonful toxin. What's the characteristic? This protein, of course, had label, but inactivated in 60 or 80 degrees. Actively secreted by cells and diffused into the surround media. Separable by cultures by physical means such as filtration. Action often enzymatic, specific pharmacological effect for each exotoxin, specific tissue affinities, highly toxic and fatal in microorganism quantities, highly antigenic properties, action specifically neutralized by antibodies and controlled by extra chromosomal gene. In this moment, controlled by beta phage. Now please look on the picture how this bacteria.
AB toxins are proteins that consist of two parts, A and B. The A portion is the enzyme that constitutes the toxic part. The B portion binds to host cell receptors. There are several mechanisms by which AB toxins enter the cell. One mechanism involves the uptake of the toxin after binding by endocytosis. After endocytosis, the contents of the vacuole become acidic, causing the A and B portions to separate. The A portion enters the cytoplasm of the cell and exerts its toxic effect, while the B portion is pushed from the cell by exocytosis. Different microorganisms produce A toxins with different activities. The A toxin produced by Corynebacterium diphtheriae carries out the transfer of ADP ribose to elongation factor 2, EF2, and thereby inhibits protein synthesis. ADP ribosylation is a common action of AB toxins.
Okay. This stuff. And my conclusion. This exotoxin consists two parts, and A and B. A is the toxic part, which diffuse surrounding media after the separate from the B part. B part for a binding of, with a receptor on the surface of the cell. How we can detect the exotoxin production in vitro cell? By Auchter-Loney reaction in Russian literature or ELEC test in English literature. What you should do? You should prepare the media consisting serum. Put filter paper in the center of media consisting the antitoxic serum. After them, you inoculate view culture from each sick person. Incubate in thermostat during 24 hours in 37 degrees. What happened during this 24 hours? Antitoxic serum diffuse through the media, and toxin, if the view culture can produce the toxin, toxin will diffuse through the media too. And in place where the toxin inactivated by intoxic serum, we can detect precipitation lines. On this picture, you can see only two toxic cultures from the sick person, or from the sick person. Now, clinical forms of Corynebacterium diphtheria. By site of infection, we can divide it in next group: facial, laryngeal, nasal, otitis, conjunctival, genital, and cutaneous. And severe of this infection from mild inflammation to hypertoxic reaction, septic form, and hemorrhagic form. On the right side of the screen, you can see the examination of throat, and you can see the fibrinous films on the tonsils. And next picture. This is picture, you can see edema, the hypertoxic reaction of this disease. Next, it's so very important, that's differential diagnostic between diphtheria and other acute respiratory tract infection which affect the tonsils. On this picture, you can see healthy throat. This is laryngitis, pharyngitis caused by respiratory viruses. You can see reddish mucous membrane and reddish tonsils. Tonsillitis is caused by bacterial infections such as streptococcal infection or staphylococcal infection. You can see cover on the tonsils and reddish mucous membrane in the throat. After diphtheria, you can see grayish or sometimes greenish films on the tonsils. And if the disease caused by fungus, you can see there's cover, but this cover is very easy to remove. In the left side, you can see the differential diagnostic table by seeing signs. Tonsils, red exudate, present other signs like the fever, dysphagia. What the causative agent are more common? After 3 years, streptococcal pathogen. Before 3 years, viral or sometimes streptococcal infection. Ulcerated necrosis.
What is this figure? Now, what is this figure? This figure, other sign, this figure.
This diphtheria, this part.
Yeah.
Next, ulcerated necrotic is caused by fusospiracetosis, and we call Vincent angina. False membrane, gray, adherens mucosa, extensive alterated general state, and very difficult to remove. If you will try to remove films in a diphtheria patient, it's very difficult because if you remember pathology, fibrinous inflammation. Fibrinous inflammation, these films connect not only mucous membrane and submucous level too. When you try to remove the film, you will detect the small drops of blood on the surface of the tonsils. Next, the principles of treatment. Diphtheria is very communicable disease. At first, what you should do, you should isolate this person, sick person, to hospital. Next step, you should add antitoxic serum, not antibiotics, antitoxic serum, because you will diphtheria produce exotoxin, very poisonful toxin. You should inactivate it, because if you will not do fast, you will see complications by this exotoxin. After that, when you introduce antitoxic serum, you should administrate antibiotics, such as penicillin groups, macrolides groups, maybe cephalosporins groups. Of course, you should notify to public health department because this is vaccine preventable disease. Next, after them, because this is toxinemic infection, you should monitoring respiratory obstruction and monitoring myocarditis. Immunity after this disease. Strong post-infection antitoxic. Carriage can be detected in 6% of this after this disease. Antitoxic immunity detection we can prepare by Schick reaction. How we prepare? Like a Mantoux test. We inject an intradermally forearm 0.2 milliliter of diphtheria toxin. After 24 or 48 hours, you should check results. You have 4 variants of results. First, if no reaction, we should check like a negative reaction. It means the immunity present. If no reaction of control, we have two arms. 4 arms tested, test 4 arms where you injected, and the control arms. If no reaction on control arms and circumscribed reaction on the test arm, you will interpret it like susceptibility test. If you detect pseudo-reaction like a diffuse erythematous reaction on the both arms, you should interpret it like hypersensitive reaction to Corynebacterium diphtheria. And combined reaction, if you detect pseudo-reaction on both arms and circumscribe it in the place of injection, you should interpret this one like a hypersensitive and susceptibility test. Specific prevention. Specific prevention in India. I found the immunization schedule in India, published in 2018 year. We should prevent diphtheria by DTaP, diphtheria, tetanus, acellular pertussis vaccine. What is the interesting? Because India immunizate only children at 6, 10, and 14 weeks. Last age period when the child can be vaccinated in 1 year old by this schedule. In our country, we vaccinate our children at 8, 12, and 14 weeks after birth, revaccinated after 14 months. And next, second revaccination will be before school at 6 or 7 years. And the last, next third revaccination by DT vaccine with the excluded pertussis at 16 years old. And we should repeat this vaccination every 10 years in 26 years old, 36 years old, and etc. Now,
Ma'am,
study. Please, I need
ma'am, I read the pertussis vaccine does not work after 1 year. It not
yes, I
have
will
the
tell in next lecture. Yes, you agree. Okay, switch on your microphones. I need your discussion. Okay. Case study. Okay. Students, switch on your microphones. I will address it, the question for one person, please. Please don't speak when your colleague will give answer. Danish Hussein.
Yes, ma'am.
Case study for all students, please read and listen. Child, 5 years old, hospitalized with high temperature, 39 degrees, mild throat pain, edema in neck area. This child is non-vaccinated. Examination of throat. Please, Danish Hussein, tell us, please describe the throat. What you see in this picture?
Ma'am, in this picture, some patches on tongue and scratch on tongue.
You can see tonsils, yes?
Yes, ma'am. Tonsils. Like we drink cold water or we take some cold thing.
Yes, you mean this cover, yes?
Yes, ma'am.
Describe what you see. How you can describe this cover? What the color?
Pink.
Maybe. Pink is mucous membrane.
Yes, ma'am.
Shivam Kurana helps to Danish.
I can see here clear inflammation of the mucous membrane.
Yes, inflammation. Okay.
And ma'am, some red patches on the tongue. And I think this disease is caused by diphtheria.
Yes, you can think.
And ma'am, also 5% to 40% of bacteriation on tongue.
But before it, you should, okay.
Edema in neck area also. Edema, some accumulation of fluid in the neck area. And some kind of mild throat pain.
How you can do differential diagnostic between pus inflammatory and fibrinose inflammatory?
Sorry, ma'am, can you repeat your question?
Shivam Kurana, can you how you will do differential diagnostic of this one? What you should examine in the throat?
Ma'am,
of course, okay. What the samples you should take for microbiological investigation?
Saliva.
Ma'am, we take the
saliva.
Not saliva. The material from the mucous membrane.
Mucous membrane. But in the merges between healthy part of tonsils and inflammatory part of tonsils. Between this part on the merges of the film. Okay. Next, for differential diagnostic, I told you that if you try to remove the film, you will, it's very difficult to remember.
First antitoxin, first we give antitoxin.
Antitoxin.
You will detect drops of blood.
Yes, ma'am.
Because you will severe damage of tonsils membrane. Okay, next question. We should, what you should exclude in differential diagnostic? Of course, you should exclude past inflammatory diseases. Next,
Viral.
Vincent angina, but you will exclude this variant on the microbiological investigation. Material for investigation starts from the mergers of healthy and inflammatory part of tonsils. Okay, Sohail, please tell us what the technique staining of the sample you will use.
Means what?
Technique staining.
Technique staining. We have to take throat liquid. Hello.
Okay, you will prepare the smear. How you color it? This
Yes, Miss by stain.
What the name of technique staining?
Yes.
Anybody have? Mubarak, maybe? I told you at the start of the lecture, show the pictures, students.
Elixir precipitation.
No, we don't use this technique.
And first we will take the materials from the board area.
You take, but you prepare after them. You prepare the smell.
When we prepare on culture and medium, like low first serum slop made, like
yes.
This type of,
Before it, you should prepare the microscopy. Before this one.
Microscopy. Before that, microscopy. Smear stain with methyl glue, and then we see under microscope.
Stop, not all students, because it's too noisy.
First we smear, the way we take from the membrane, mucous membrane.
Yes, you took it.
Specimen. Then we this smear is stained with methyl, and then we take seen under microscope.
Yes. Before it, what's the method of staining you will use?
And then after
throat swab.
Staining.
Ma'am, we have to stain from low-flare methylene
throat swab method.
Low-flare, yes, low-flare technique.
Methylene blue.
Serum slow.
And
telluride blood agar.
No.
Rapid strep test.
No, some later when you will have a pew culture.
Blood agar.
Before agar.
Blood agar.
Blood agar we use for cultivation. You have smear on the slide. You should stain the slide. Okay, first answer was Loeffler's technique staining. I'm agree. Second technique staining is Albert Neisser.
Albert Neisser, yes, ma'am.
Because by first microscopy technique, you can differentiate this disease from the fast inflammatory, because you should detect metachromatic granules. Okay, after them, you inoculate on the enrichment media. And names of the media alarms say told to us. Shahrukh, please read.
Yeah, local serum and telluride, blood agar.
Blood agar, okay.
Yeah, I repeat.
Telluride, blood agar,
Yeah, blood
and
agar, low flood serum, telluride, blood agar. Blood agar and telluride, blood agar, because telluride we add on blood agar because telluride is not allow the development of other flora of mucous membrane. Sorry.
Oh, sorry. Oh.
And your sound is not coming.
Sorry.
we can use metalloid medium.
Wait one moment. Okay, microbiology investigation. You will use conclusion. Blood agar consisting telluride. Okay. What you should do with this patient?
For treatment.
Stop. This patient will do personality for reticum.
Yes, miss.
What you should do with this person, firstly? You will think about diphtheria disease.
This disease are transmitted person to person.
Not transmitted. Urvi?
अरे मैंने एक उदाहरण लिखा था उसे ही मैम बोल रही थी तो गूगल में सर्च करना भेजा हूं तेरे को तेरे को भेजा हूं ना डार्क स्टूड जो है ना मिटा होगा वही भेजा है
Students, please don't disturb it.
हाँ तीनों
Margu.
Ada biari tu poda alam.
क्वेश्चन में दो क्वेश्चन एक क्लासरूम और डार्क स्टूड में क्वेश्चन
Okay, Irfan, what you will do with this sick person?
Sick person, if he has inflammation, then we should take take saliva and we should test. And if he has like edema and inflammation on tongue, then we should treatment this person
Okay,
by
how you will treat?
By medicine and some prevention like cold, drink cold and boil water, not very hot because it has patches on tongue. And
okay, what the treatment you will use?
We should give antitoxins and antibacterial.
First we have to isolate this person.
After them, you will give antitoxic serum. I'm agree with you. After them, what you should administer? What you should give to the person?
Antibiotics.
Yes.
Antibiotics. And
after that, we have to notify the public health department.
Of course.
And ma'am, after that, we have to inspection or monitor of respiratory conditions of the person. And ma'am, after that, maybe in some cases endocarditis.
Yes.
Yes, ma'am.
Okay. Of course, this child lives with parents, and they have close contact with these parents, and maybe brother and sister. What you will do with person who was in close contact?
Ma'am, first we have to isolate this person.
Yes.
Because this disease is transmitted from person to person.
Yes. Put in quarantine. Yes?
Yes, ma'am.
Yes, ma'am.
Okay. After them, maybe you should detect this person are infected by Corynebacteria diphtheria or not. And you should take the samples and incubate on the thermostat and check results. Okay, after 48 hours, we have results of bacterial investigation. You will see this microscopy result and a result on the blood telluride media. What you can tell about this? Sorry, this 3 contact persons. This result by microscopy. What you can tell?
Ma'am, by microscopy, there is a presence of chains or something like bubbles.
Yes, this purple color because this Loeffler medium or Loeffler staining. Sorry.
And ma'am, in telluride blood agar medium, we can see some granules, which we call volutin granules.
Volutin granules you can see only in microscopy. Here,
okay.
Yes, in the end of the rods.
Yes, ma'am.
Next, how you can describe colonies on the telluride medium?
Color.
Some dark round colonies.
Yes, dark, brown.
Round in shape, ma'am.
By the, I can help you. This colonies I demonstrated in our lecture from the biovars. You can think about some biovar of Corynebacterium diphtheria.
Sorry, ma'am, can you please repeat?
This the colonies of on this media I demonstrated when I described the biovars of Corynebacteria diphtheria. This colonies looks like a daisy head. What's the biovar of it? Shivam?
Yes, ma'am.
Maybe you know the answer. Okay, Akanksha. This is daisy head.
Yes, ma'am.
This is Gravis biovar. Students. Okay, next you prepare the biochemical test and results of the PISU test from the sick person and 4 contact persons. What you can see here, this positive or negative results?
Positive result.
Positive, where is positive?
Ma'am, where we can see black film.
Machine fourth,
First, second, and third.
Yes, sick person.
But but 4% is not much more affected.
Fourth one is clearly treated
And not much more affected.
Okay, who told about sick person and for first, second, and third person are positive for PISU test? We can detect presence of testinase here because we can see black
Black color. Yes, ma'am.
along the injection in this
Yes, ma'am.
inoculation. And fourth person are negative for PISU test.
Not seen. Negative.
Next, result of Zach's test. What you can see. Where's positive test?
Say the first, second, and third.
At the third
Pink color.
Pink color.
Positive
Pink
is
color
yellow one.
is positive, or yellow color
Yes,
is
ma'am.
positive.
Yellow, ma'am.
Yes, yellow color is
Yellow, ma'am.
color is positive.
Ma'am,
Yes,
yellow
yellow color
positive.
is positive, and
Positive.
positive Zach's test is characterized for what kind of Corynebacteria? Sick person, first, second, and third contact person are negative for Zach's test, and fourth person is positive for Zach's test. And if you remember, PISU test is negative for fourth person.
Ma'am, fourth person will be like Corynebacterium diphtheria because it shows negative impact from urease.
Yes, and if Corynebacteria doesn't produce urease, they don't. This bacteria doesn't change the pH.
Yes, ma'am. Color will not change.
Yes, and first, sick person and 3 contact persons has
positive result, so it will show
diphtheria, positive
change
result
in color.
diphtheria by negative Zach's test. And fourth person
Change
are
in color.
positive for Zach's test. We can think about diphtheroids, non-pathogenic Corynebacteria.
Non-pathogenic forms, yes, ma'am.
Next, we should detect this bacteria is has toxigenicity or not. We will do next test. What the test we will do?
Ma'am, ELEC test?
Yes, ELEC test. This ELEC test. And check results.
Ma'am, first, second, third, and fourth shows positive result.
They have
Fifth
Pew culture produces the toxin, and this toxin is are inactivated by antitoxic serum.
Yeah.
Yes, ma'am.
And fourth person has non-toxigenic culture. And we are sure that fourth person has diphtheroids or non-pathogenic Corynebacteria, commensal flora of our nose or throat. Okay, what you will recommend to person from contact?
Okay.
What you will recommend to person from contact?
People should not attach with someone and live with in isolation.
Okay, you will isolate it, put in quarantine. I'm agree.
Yes,
Next, but you detect the Corynebacteria diphtheria toxigenic strain in 3% from the from the 4. 3% are carriers.
And we can use specific prevention.
Yes, you may, but before the specific prevention, what you should prepare?
For detection of disease in a patient.
Then we can
You should prepare
immunity
test.
antitoxin.
And of course, you should prepare emergency vaccination against diphtheria. Okay, next, possible complication of disease. What we should afraid?
First one is blocking of airways. Can be.
What's the complication of this disease?
And blockage of air passages.
Eating food problem.
Like
Problem
for
in digestion.
Formation of mucus
asphyxia. Firstly. You told us that what you will do in treatment tactic.
Treatment, we can use specific prevention like vaccine.
Yes, of course, after prevention. Complications.
Complications, ma'am, basically symptoms, huh?
Okay, so what the kind of symptoms you will detect in complication?
And blockage of air passages like pharynx.
Nasal.
What you will monitor, monitoring of function, what the kind of organs you will monitoring?
Nose, Miss Nose.
Nose, okay. And
pharynx.
Throat upper part.
Oral cavity.
Upper respiratory region, ma'am.
Miss oral cavity.
Tract.
Yeah. Conjunctival, man, like eye area. In complication,
Function of heart muscle and respiratory obstruction. Okay. We will complete discussion by case study of